We're back with Mendelian genetics. Now we're going to look at dihybrid crosses. So in these, now we have two different alleles represented by two separate letters. Two different separate letters in the cross. So we're going to look at pea pods. The big T is going to be tall. Little t is going to be short plants. And L is going to, let's say L is for normal leaves. And little l is going to be for wrinkled. And in this cross, we're going to take two parents. One of them is going to be heterozygous for height and homozygous recessive for leaves times dom for height. And let's make a heterozygous for leaves. So we have here, we're going to 4 by 4, so we're going to have 16. How are we going to get to that many? So what we're going to use is FOIL. Like in math, we're going to take first, outer, inside, left. And so we're going to get our combinations here. So big T, little l, big T, little l, T, little l. And you can see I'm just going like this, then I'm going here, then I'm going like that, and then I'm going like that. T, little l. Okay, so we got all the ones there. Now we're going to go to these. We're going to go that, to that, to that, and then to that. FOIL. Okay, so we have T, L, T, little l, T, L, and again, T, little l. As we could see here, we would have 16 by 16. 16 boxes because it's a 4 by 4. Sorry about that. And now we could just carry on the letters like we did the monohybrid. So we're going to have big T's here. All big T's. And going to have alternating L's. It's going to take a while. And there's some patterns you can notice with these too. So once we finish this one off, we can look at some of the patterns. Okay, now we're on the little t's. You really want to exaggerate the letters here. Again, more little t's. We have all big T's going across here. These crosses. We have all little t's down, and we have alternating in here. Yep. Okay, so we finished all our crosses. I don't think we messed up. That's a big T. Just need to make note about that. There we go. Okay, so now we look at our boxes. We have 16 possible squares. Offspring. Just touch up a few errors here. Okay, so we could see here, this bottom, we have TL, big L, T, little l, and TL and T, little l again here. We also have repeat. Do we have repeats? We have repeats up. We don't have repeats up there, so actually the trick won't work. Oh wait, yeah it would. Okay, so do you see how these two columns are the same as these two columns? Well, could have just done, we could disregard this area now, and if we see this pattern here where these two are copies of these two, then we could just discard this log bar, and we would just have a 2 by 4 over here. So look at the same ratios. That's only if these set here a repeat of this upper set over there. And so now I'm going to look at for phenotypes. So we have all of them are tall. all of them are tall, so all out of 8 are tall. 8 out of 8 tall. And we do have some carriers. We have some over here. So they're heterozygous for height. All out of 4 out of all of them, so 1/2 are gonna be 1/2 of them are gonna be tall and normal. The other half are gonna be tall and wrinkled. And we're just counting all the things. If we were looking at genotypes, we'd have to go through all of them, make a chart with the percentages. That's Dihybrids.